Good afternoon and welcome to the second of 3 anniversary symposia for the MassCPR. Thank you very much for joining us this afternoon. Today's symposia will be on the very important topics of COVID-19 vaccines and pathogenesis. Over the past 12 to 14 months, there has been astounding progress in the development of vaccines and our understanding of pathogenesis for this viral pandemic. We have not 1, not 2, but 3 approved vaccines under emergency use authorization that are currently being rolled out on a massive scale in the United States and throughout the world. It's truly a stunning accomplishment of science. And for pathogenesis, we've never seen such rapid progress in our understanding of the virology, immunology, and pathogenesis of this disease. So we're delighted to bring 5 outstanding talks to you today to give an update on this very important topic. Galit?
Thanks, Dan. So it is my pleasure to introduce our 5 outstanding speakers for this session on vaccines and pathogenesis. Our first speaker is Andrea Carfi, Vice President and Head of Research on Infectious Diseases at Moderna. Now, Andrea has been at the leading edge of structure-guided vaccine development for decades and has been instrumental in driving the Moderna mRNA vaccine program from design to clinical deployment. Our second speaker is Stephenson, an assistant professor at Harvard Medical School and the director of the clinical trials unit at the Center for Virology and Vaccine Research at the BIDMC. Dr. Stephenson is a physician scientist who specializes in the development of preclinical and clinical studies, and she has been instrumental in testing the safety and immunogenicity of the Johnson & Johnson vaccine. Our third speaker is Nir Hacohen, who is a professor of medicine at Harvard Medical School and an institute member at the Broad. Nir is the co-director of the Cell Circuits program at the Broad, and Nir has been rewriting the book on immunology, one disease at a time. And today, Nir will be telling us about his unexpected discoveries related to pathological cells in COVID-19. Our fourth speaker is Amanda Martinot, an assistant professor at the Cummings School of Veterinary Medicine at Tufts. Mandy is a veterinary pathologist specializing in animal models of human infectious disease, and her lab primarily focuses on the immunopathogenesis of infectious diseases. And she'll be sharing some of her fascinating discoveries from her COVID-19 work. Our final speaker is Pardis, who is a professor of medicine at Harvard Medical School and an institute member at the Broad. Pardis has been front and center in the battle against many pandemics and epidemics, helping to push our understanding and awareness of pathogen evolution to fully deploy countermeasures to fight back against pathogens. Now, before I turn this over to the speakers, I'd like to remind the audience to put their questions in the Q&A box, and Dan and I will pick them out and make sure to address as many of them as possible with the speakers at the end of the session. Now, with that, I will turn the podium over to our first speaker, Dr. Carfi. Take it away.
Thank you, Galit. I'm going to share my screen. So I will skip the disclaimer slide and move directly to the work that we have performed on the development of the Moderna COVID-19 vaccine. The vaccine consists of a messenger RNA encapsulated in a lipid nanoparticle that is delivered through an intramuscular injection. Then the mRNA LNP migrates to the draining lymph nodes where it transfects antigen-presented cells. Once the mRNA is within the cells, of course, engage the ribosome and results in protein expression. In this case, the spike protein from the SARS-CoV-2 virus that has been stabilized by the introduction of 2 protein mutations. Of course, the spike protein is seen by the immune system, resulting in a strong adaptive immune response. The development of the vaccine was fast, and this is thanks to the technology, but also to the collaboration with NIH, BARDA, Operation Warp Speed, and of course, the help of the regulatory agencies. We started at the beginning of January when the sequence of the virus became available. Within 63 days, we were in a phase 1 study sponsored by NIH. End of July, we started a phase 3 study that was an efficacy study. And by end of November, basically, we had the initial efficacy data, which allowed for an emergency use authorization by mid of December. So in 11 months, we went from sequence to an authorized vaccine under EUA. So we performed, of course, preclinical experiments at the same time, and in both monkeys and hamsters. Here are data from monkeys to show that the vaccine can prevent infection. Animals received the 2 doses of the vaccine and then were challenged. And we could see that after the challenge, by looking at by PCR replicating virus, we see that the vaccine basically protects. There's no replicating virus in the 100 microgram dose in the bowel, but also in the right graph, also in the nose. At day 2, there is no animals with the replicating virus and only one replicating virus at day 4. So the immunoresponses elicited by the vaccine are really able to control the virus very quickly and are protective in the lung and partially protective in the nose. So in parallel with the non-clinical studies, we conducted a number of clinical studies, a phase 1 to demonstrate safety immunogenicity, followed by a phase 2 study to confirm the dose, and then a phase 3 efficacy study. Currently, we are conducting additional studies to assess immunogenicity and also safety of the vaccine in different populations, among which are the pediatric and also immunocompromised populations. The phase 3 study was a randomized placebo-controlled safety and efficacy study involving like 30,000 subjects. Half of them received a saline as a placebo, basically, and then the other half, the vaccine at the 100 microgram dose with 2 injections 1 month apart. The primary endpoint was prevention of symptomatic COVID-19, but we also looked, of course, at prevention of severe disease and also infection as a secondary endpoint, among others. And really the primary efficacy analysis demonstrated that the vaccine is highly efficacious with a 94% efficacy. At the time of the efficacy analysis, we had 185 cases in the placebo group and only 11 in the vaccinated group. I mentioned that we looked also severe disease, and at the time of the primary efficacy, we had 30 cases in the placebo group, but none in the vaccinated group, pointing to 100% efficacy in prevention of a severe disease. When we look at the Kaplan-Meier curves, we can see that there is a divergence of the curves for the placebo and the group that received the vaccine, and the curves start to separate even before the subjects receive the second dose, suggesting that the vaccine can actually protect even after 1 dose. And this is being confirmed now by real world data, showing that the vaccine is also protective after 1 dose and also protect in terms of prevention of infection. So more recently, we have been working on the variants of concerns. Pardis will tell us more about that. There are a number of variant viruses that are being identified in different parts of the world, and they are spreading across the world. The 117 that was identified first in UK, the 351 in South Africa, P1 in Brazil, and the 429 in California. They have a number of mutations, but it's important to point out that some of these are in the receptor binding domain and the N-terminal domain, which are the targets for neutralizing antibodies. So this mutation may have an impact on the vaccine potency and efficacy. So we evaluated the sera from subjects that received the vaccine in phase 1 using a pseudovirus neutralization assay against these variants. And what we could see that there's not much of an impact on neutralization when the 117 variant is tested, but we see an impact in the potency of the neutralization when the other variants are tested, going from 2.3 to 6.4 fold decrease. However, I would like to highlight that the neutralizing titers are still above 1 to 300, that are levels that we know are protective in monkeys, challenged with the virus. So we think the vaccine is still protective, but there might be an impact on the duration of protection. For these reasons, we are evaluating strategies really to improve immunogenicity with a 3rd dose. And the approaches that we are testing preclinically and now also in the clinic is a 3rd dose of the Moderna COVID-19 vaccine, 1273, a variant-specific booster candidate that is based on the sequence of the variant that started in South Africa, a multivalent booster candidate that is a mix of the 1273 and the 351. And finally, for subjects that have not received the vaccine, we are also testing a primary series of the 351 or the 211. So these studies are ongoing, and we should have data in the next month. So I would like to finish by acknowledging that this was a really large collaboration. We had really help from NIH, BARDA, Operation Warp Speed, and I would like to thank the investigators and also all the people that participated to the clinical studies. So with that, I will stop and I'll pass the baton to Katy Stephenson that will tell us more about the adeno approach. Thanks.
Thank you, Dr. Carfi. All right, let me just share. Okay, I'm going to get started. All right, thank you for that wonderful presentation about the mRNA vaccine. I'm going to turn now and talk about the Johnson & Johnson Ad26 vaccine. So this is a different technology than what you were just hearing about. This vaccine is based on a replication incompetent adenovirus 26 vector that delivers a transgene that encodes the spike protein. So this virus vector that is used to deliver the protein started out as just a regular old wild type adenovirus 26, and this virus does circulate among humans but doesn't cause clinically significant illness. But what's done to it is that it is modified. So a number of genes are deleted from this virus, and it renders the virus incompetent, meaning it can't replicate anymore. So it can get into cells and deliver its cargo, but it can't get out of the cell. This replication incompetent vector now also has space to add a transgene. And so this schema at the bottom just shows the transgene for the spike protein. And it encodes the full-length spike protein, and it only has a couple of mutations that stabilize the protein. So Dr. Dan Barouch led the effort at the Center for Virology and Vaccine Research at BIDMC to begin the development of this vaccine, which started back in January of 2020. Our group then partnered with Johnson & Johnson in the spring, and we've worked with them before for Zika and HIV, so it was a natural partnership. And we began the first in human trial, the phase 1/2 trial of this vaccine in July of 2020. And that study was focused on safety, of course, but also really on dose selection. So trying to get to that point where we could pick something out that was going to go into the phase 3 trial. And that was done. That was done actually by September, basically, very fast. And that's when the phase 3 COVID-3 trial began. So I'm going to just show a couple of slides from the data that we found from our phase 1 trial, mostly because one of the most common questions that I get about the Johnson & Johnson vaccine is, how did they decide to just use one dose? Because it's the only one out there that's doing that. So where does that come from? And part of the answer to that is from the data from this early study. So in our early study, we tested 2 doses of the vaccine. The first is 5 × 10⁻10 viral particles. It's actually more or less a standard dose because that's what's being tried in a lot of other studies. Versus a high dose of 1 × 10⁻11. And then we also looked at 2 doses versus 1. So this is a figure that shows the peak antibody responses. I've highlighted those peak responses with these rectangles. And what I really want to just show is that at those peak responses, which were 71 days following immunization, the standard dose after a single immunization was essentially the same peak responses as all the rest. So, and particularly 1 dose versus 2 doses was definitely not different. So this was also borne out with a look at the T-cell responses, which were pretty robust at 71 days. And again, peak responses that were observed for the standard dose just given 1 time were definitely not inferior to the other regimens. So that was in fact what was selected to go forward into the ensemble trial. So in the ensemble study, there were a big bucket of people, 40,000 people, and they were randomly assigned to either receive the single injection of the vaccine or a single injection of placebo. And the way these studies work is that, we advise everybody, like, wear your mask, do social distancing, do everything you can to protect yourself, but we're still going to monitor you for infections that occur. So in this trial, ultimately they collected about 400 or so cases. And at that point, they were able to open the books and look at the data. So this study was conducted in North America and Central South America, and then also South Africa. But I'm just going to show you a quick snapshot of the demographics of the study here in the United States. And they really did a fantastic job recruiting a population of people that represent the diversity of the United States. So it was about 50-50 men and women. A good quarter of the study population was greater than 65 years old, which is fantastic. 15% of the participants were Hispanic, and this is really good for a clinical trial. 13% of the participants identified as Black or African American. So now we get into the primary data. What did they find? So looking globally, all of those different countries that they went to, they collected 437 cases. And of those cases, 324 were in the placebo group and 113 were in the vaccine group. And this is, when I say cases, I mean symptomatic cases that were at least moderate illness. This calculates out to an efficacy of 65.5%. Now, if we look further just at our own United States, the data is a little bit better. So there were 144 cases in the US, and only 32 of those cases were in the vaccine group. So that calculates to 72% efficacy. Really importantly, though, is efficacy against severe disease, because I think all of us, of course, no one wants to get sick, and there are consequences of even moderate illness, but we also don't want to get into the hospital or the ICU. So one of the findings from this is that the efficacy was very good against severe or critical COVID-19. So here in the US, it was 85.9% effective. And in the data, nothing's 100%, of course, but it was near 100%, as you can see in a study, against COVID that required a big intervention, like being admitted to a hospital or being on a ventilator or death. So an excellent protection against those events. This is a figure that shows the separation of those events. So in the placebo group, which is in gray, is the cumulative incidence of cases of severe disease, versus in the vaccine group, there's a separation of, and what's really interesting is that you actually start to see that separation only 1 week after a single shot of the vaccine. We're worried these days about our variants of concern, and Dr. Carfi outlined some of those. This is the data that shows the protection against severe disease that was analyzed by country. So I mentioned that this study was done in all these different countries, and coincidentally that happened to take place at exactly the same time that these variants of concern were being detected within these countries. So in particular in South Africa, it was really interesting to observe that 82% protection was seen against severe disease, even in the context of there being more than 90% of the cases, the 351 variant, which is one of the more concerning of the variants. So let me summarize here and just include a few additional features and findings about this vaccine. So it has been safe and well tolerated in all of the studies so far. Interestingly, the breakthrough infections that were detected among people who received the vaccine appear to be less symptomatic than you would expect otherwise, which is great. There's evidence of protection against asymptomatic infection. The shipping and storage requirements for this vaccine are favorable, and they fit into the existing medical supply infrastructure. And the single dose regimen allows rapid vaccination of a population. And this is not the end of the data. There's a second study, Ensemble 2, which is ongoing, which is going to be testing, or is testing, the 2-dose regimen so that we can actually find out if 2 doses is better than 1. And I'll just say, with all these acknowledgements, I just want to highlight in particular the clinical trial participants, who are my beloved heroes. Thank you. Oh, so now next up we have Dr. Hacohen. Excuse me, Hacohen, sorry, Nir, and he will begin his presentation. Thank you.
Fantastic, thanks. That was so exciting to hear about both vaccines. Okay, so what I'm going to talk about is the pathogenesis of COVID, and this is carried out by a large group across Mass General, Brigham, Broad Institute, and MIT. And a lot of it is based on this cohort that we collected during the peak time around March, April at MGH of an emergency department cohort collected by Mike Philbin, a physician at the emergency department, that covered really all the different levels of severity of COVID, from death to being discharged in the emergency room, with about 306 patients and 78 controls. And this collection allowed us to study the immune correlates of severity, to look at plasma proteins, transcriptomes of millions of cells, antibody responses, viral load, working with many teams across MassCPR. And so the story I want to tell you today really comes from an initial study that we did together with the same group at MGH on sepsis that we published just last year, where we looked at the single cell genomics of bacterial sepsis, in particular urinary tract infections, and identified an immune cell, a monocyte cell that we call MS1, myeloid state 1, which is found in strong association with bacterial sepsis in this cohort. And so here you can see on the right that healthy patients have almost none of these MS1 monocytes, but the septic patients have very high levels. So we wondered if this might occur as well in COVID. So we looked across multiple cohorts, actually, not just ours, and we found that in severe COVID, in fact, there was much higher levels of this pathogenic cell, this MS1 cell, in every cohort we could look at. These are just some of the genes that are the markers of this particular cell type, if anyone's interested. And so it looked like it was very general, both bacterial sepsis and COVID. And so next thing we wondered is, where did these cells come from? Because our initial data suggested they don't come from the standard cells that are in the blood, but rather maybe they come from the bone marrow where cells form initially, the immune cells. So we wondered if we took plasma or blood from patients with sepsis or severe COVID-19 and simply added it to cells from a healthy person's donor bone marrow, where all the progenitors and stem cells are. And in fact, when we did that, and not with healthy or mild COVID or infectious plasma, only with severe COVID, we were able to indeed generate this MS1 signature, shown here, the MS1 score, both in monocytes as well as in neutrophils. And these are very similar to myeloid-derived suppressor cells that have been described before. So we're able to generate them directly from bone marrow with plasma. And we wondered, what do these cells do? What are their functions? They're in really high levels in these severe patients. Do they impact our sensing of pathogens? Do they impact the activation of T cells? So first thing we looked at is pathogen sensing, and we stimulated cells with RNA to mimic a virus. And what we found was when we added MS1 cells to these cells that were stimulated, we had a much lower response than when we just added standard monocytes that are present in a normal person. And so we're able to suppress the RNA response, but not the response to interferon. And interestingly, if we look in the patients directly in severe COVID-19, we find that the interferon response in patients, which is what's the response to RNA, basically is lower the more MS1 cells there are in these patients. So both in vitro and in vivo, we believe this is a very important mechanism of suppressing innate immunity. We also looked at T cells. Here we activated T cells and we added these MS1 cells again and asked, do the T cells proliferate? Normally the T cells to the left here, these are when they proliferate, when we activate them. And this is non-activated T cells. So you can see when you add the MS1 cells, there's much less proliferation, almost to the amount when you don't activate them. So it's a very, very powerful suppressor of T cell proliferation. Interestingly, when we go in vivo, again, back into the both sepsis and COVID-19 cohorts, we see that the level of the MS1 cells is inversely proportional to the CD4s. And as many people know, both in sepsis and in COVID-19, we see a lymphopenia or a reduction in the CD4 T cells, which may account for a lot of important biological changes and immunological protection that occurs in patients with COVID-19. So the MS1 cell seems to both suppress the innate immune system, suppress the adaptive, and be negatively correlated with those in vivo. Working with Pravesh Khatri at Stanford, we wondered how general this is. And so he had collected from many sources almost 5,000 samples where he could look at the blood samples in patients with severe infections that had gone from mild to moderate, serious, critical, and fatal. And this is in a whole variety of infections across the world. And what we found is when we looked at our signature of 30 genes from the MS1 cell type in the blood cells of these patients, there was a very, very strong correlation with a p-value of 10 to the minus 16 of association between the MS1 signature and the seriousness of the infectious disease. And when we look at individual viruses, you can see for influenza, rhinovirus, SARS, of course, Ebola, et cetera, all of them, there was an association with severity of infection and ultimate symptoms with the MS1 signature. So to conclude, we found that these MS1 monocytes are associated with both bacterial sepsis and severe COVID-19. They represent actually a very strong and excellent marker of severe and fatal infections of any kind. We found that the plasma factors from these severe patients can induce these MS1 cells directly from healthy bone marrow, and that these cells are immunosuppressive for both innate and adaptive immunity. These MS1 cells are very similar to what previously have been found by Dmitry Garbilovich originally and others, called the myeloid-derived suppressor cells, and they represent a powerful negative feedback that can suppress the immune system in these severe situations. We believe that in many cases, these cells are actually helping to block some of the increased cytokine storms that we see that lead, in fact, to more fatal disease. So they are a negative feedback system. And so we're now trying to figure out how to manipulate them and use them for pathogenesis. Thanks so much. And our next speaker is going to be Amanda Martinot.
Thank you, Nir. That was great. Okay. Good afternoon, everyone. Today, I'll be sharing with you how we can use animal models to teach us about COVID-19 infection. So SARS-CoV-2 infection leads to a type of pneumonia called interstitial pneumonia that leads to, in severe cases, acute respiratory distress syndrome, or ARDS. Now, in a normal interstitium should only be 2 cells thick. That should be enough space for a red blood cell to interact with the pneumocyte to allow the proper exchange of oxygen and carbon dioxide. But in SARS-CoV-2 infection, as seen with SARS and MERS and other coronaviral infections, the virus infects the respiratory epithelium of the lower airways, and this leads to a rapid inflammation and arrival of neutrophils from the bloodstream, that it goes to the interstitial space and into the airspace. And the combination of the viral replication and this inflammatory response really leads to damage and loss of the airway epithelium and replacement by a structure called hyaline membranes, which is a pathognomonic feature of ARDS pathology. Now, in human medicine, there are 2 phases of ARDS described. There's an acute phase and an organizing phase. And the organizing phase is characterized by the pathology of repair, which is seen with type 2 pneumocyte hyperplasia, the proliferation of fibroblasts, which can lead to fibrosis and loss of lung function, and ultimately drainage of edema and restoration of lung function. Unfortunately, in the early days of the COVID-19 pandemic, we really only had a window into this later phase of disease because the only specimens available to us were autopsy specimens from terminal patients, which is why we really needed animal models to try to understand this earlier phase of disease and try to figure out ways that we could intervene and prevent long-term effects. So what do we see in autopsy specimens from human patients? These are some ultrasound-guided autopsies that were performed at Beth Israel Deaconess Medical Center. And again, we can see the classic ARDS pathology with the formation of these hyaline membranes, which are essentially just a protein covering over an area of damage in the lung. And this overlaps with areas of ongoing viral replication, as seen by viral protein here and viral RNA. But again, these autopsy specimens really limited our ability to understand and distinguish what was happening in COVID-19 from other coronaviral infections, and we really needed animal models to better understand what was happening in this virus. Fortunately, we had the opportunity to collaborate with Dan Barouch at Beth Israel Deaconess Medical Center, who had a massive vaccine program, as you know, and we tested a number of animal models trying to find ones that would best recapitulate what was described in human patients. And there was a lot of expectation for the non-human primate model, especially for vaccine development. So how well does the non-human primate model recapitulate human disease? Despite the monkeys actually having very, very mild to no clinical disease, we could find evidence of epithelial damage in the lungs of these monkeys that definitely recapitulated what was seen in human pathology. So you can see that the virus does infect the respiratory epithelium. This does lead to damage and fluid accumulation in the lungs of these monkeys, with attempts of the epithelium to repair itself with proliferation and interstitial cell formation, and ultimately repair by the presence of hyaline membranes. One of the really striking features of the infection in monkeys was this really massive infiltration of macrophages and neutrophils very, very early in infection at 2 days post-infection, that then was rapidly resolving, which likely explains why the monkeys don't develop severe clinical disease. But despite that, we do find evidence of long-term reparative effects in the monkeys, the lungs of these monkeys out to 10 days post-infection, which again models probably what's happening in mild disease in people. So again, here you can see an example of a normal alveolar space with, again, with that 2-cell thick, a red blood cell and a pneumocyte in a normal region of a lung, while in these affected regions of the lung, you can see that there's far too many cells here, and this is preventing normal oxygen exchange. And these cells are representative of epithelial cells proliferating and also macrophages is infiltrating. We can see other evidence of damage in the reparative process, or the sort of organizing phase. These syncytial cells, again, too many nuclei in this cell. The pneumocytes are trying to repair, they're hyperplastic, and in some cases, they're actually promoting ongoing viral replication. So really, monkeys represent what we think is probably happening in mild to asymptomatic cases in people, where only very small portions of the lungs are affected, not enough to make the animal overtly sick. But what we really wanted to understand was how COVID-19 was different than other coronavirus infections. And one of the features that had emerged from these autopsy studies was that people were having massive thrombosis occurring in the lung and other parts of the body. And so one of the things that had been reported in autopsy specimens was that in human patients, there was the evidence of inflammation of blood vessels and thrombi formation in the lungs. So here you can see in a human autopsy specimen that there are too many cells here in this endothelial space in the vessel. And this is an evidence of activation, and we can also appreciate this in the lungs of the monkey specimens that we evaluated. And this is important because activated and inflamed vessels actually predispose to thrombus formation. So we went looking for other evidence of an early thrombotic state in the monkeys. And again, we could see the presence of fibrin, platelets, and red blood cells aggregating together, which is an early clot formation with these special stains. And this mirrored what we could see in the human lung. And we could also find evidence of these microthrombi that had been reported in the human autopsies that were in these very small vessels in the interstitial space, and also some examples of some more mature thrombi later on in disease. Most importantly, pathological assessments in non-human primates have allowed us to evaluate the efficacy of vaccines. And this is some data from the Johnson & Johnson vaccine from Dr. Barouch's lab. And so again, you can appreciate that in these animals that are non-vaccinated, in regions of the lung, there's massive inflammation, while in vaccinated animals, this is greatly, greatly reduced to none. In contrast, the hamster model is quite different. It actually is a model of severe disease. And in hamsters, they do develop clinical symptoms. They have dramatic weight loss that does recover over time. And the hamsters have a very similar pathogenesis in the sense they have massive interstitial pneumonia. It's just that the scope and degree of that pneumonia is much more severe, leading to their clinical demise. So here you can see that they have necrosis in the epithelium, again, a massive viral pneumonia, and this is representative of massive viral replication and inflammatory cell infiltrates, macrophages, and T cells. So in summary, the animal models have been absolutely critical to our understanding of early events in SARS-CoV-2 pathogenesis. The non-human primate model very well recapitulates key features of SARS-CoV-2 in human patients, including these vascular changes and a hyperthrombotic state. And it's been extremely useful for modeling mild SARS-CoV-2 disease, the immunology of infection, and also vaccine efficacy. While the hamster model will likely continue to be an important model for severe pathological outcomes and possibly for use in testing of vaccines and therapeutic interventions. And I'd like to thank Dan for the opportunity to do this work and all the members of my team who have helped. Thank you. And I will pass it on to Pardis.
Oops. All right. I think you have to unshare for me to share. Yeah, perfect. Okay, thanks so much. All right, there we go. Okay. Share this desktop. Okay. Oops, and there we go. All right, well, thank you so much. It's quite a lineup to be able to have to present at the end of, but it's my pleasure to talk at the end of this, after we talk about all the really important advances that we've had in vaccines and countermeasures, to think about a little bit, to spend the last moment and think about whether or not this is the end. And I think some of the talks have already talked about these emerging variants, but I think one of the greatest risks to our success in responding to SARS-CoV-2 are these emerging variants. Oops. And one of the things that just is important to kind of just orient ourselves is this is not a surprise. We know viruses mutate. That's, all organisms are mutating at some level. Viruses have very high mutation rates and are replicating and transmitting very quickly. And so we, in every outbreak, have seen mutations emerge, and the more genomic sequencing data we have, the more we see of that. And nowhere else have we really gotten to see this in real time the way we have it with COVID, where there's genome sequences being reported all around the world. During the Ebola outbreak, Jeremy Lubin and others were able to show how Ebola was mutating and how those mutations rose to prevalence quickly and had biological effects. Now you have the whole world looking at this and much more data. And so we see this happening, but it was likely happening many times before. The first variant of concern that kind of emerged during SARS-CoV-2 was this spike protein mutation, D614G. And what sort of set everything in motion, I remember the phone call that Jeremy called me in February, where he said, hey, there's this new mutation in Europe and Italy, and it scares me because it looks like it's in the spike protein in a place that would be biologically important, and it's starting to creep up. And he started to do those experiments then, and over the course of the months as he was doing these experiments, and other people had also taken note and started doing the experiments, this mutation really took over and almost rose to fixation. So we're really seeing evolution in real time. Jeremy and colleagues essentially were able to show that this D614G mutation looks like it increases infectivity, changes the protein structure, and there's a lot more data coming out about it. So that was really the first of it. And then began the hunt, where everyone started looking to see what else might be rising. And a number of different variants emerged. Variants that we call variants of concern because they look like they're more infectious, more deadly, might permit reinfection or render vaccines less effective. And we're seeing some of that data now, and it's good to see that it's that there is a robustness to them, but we need to stay vigilant, and there always may be new variants. I note here, it's important not to locate them with the location, like in the sense that the variants you've heard about, B117, P1, and B1351, have been called the UK variant, Brazil or South Africa. But one thing that's important to note is this is where they were discovered, not necessarily where they were born. And in fact, this should tell you that we have good genomic surveillance, because essentially everywhere that developed a good genomic surveillance system identified these variants emerging. So that's really about not COVID emergence of variants, but COVID detection of emergence of variants. That all said, we'll keep them to the names B117, P1, and P1351. These are kind of some of the big variants of concerns that are emerging, and there are other ones. And the particular mutations, so that these different lineages have a host of different mutations that we are concerned about. So when you hear about this lineage, there's a lot of different mutations on it that are marking it, including all of them starting with the D614G, since that rose to fixation. And the particular mutations that we're really concerned about are these ones that also are in receptor binding domain. So we really do think they might affect our countermeasures. And one of the reasons why there is the California now variant, but genomic surveillance in the United States has fallen behind, and that's really because of a federal funding gap. The U.S. ranks 43rd worldwide in sequencing per capita per COVID sequences. And that's not because we don't have experts here, but because the US has not prioritized genomic surveillance the way that the UK has and other countries have. That is now changing. There's an understanding that we need this, and now almost $2 billion has been put into the CDC to try to ramp this up. The US is ramping up, the Northeast is ramping up as well, and we're seeing a lot of different things in this data. Things that we're seeing is higher density of sampling in recent times. So this is a phylogenetic tree. The more variants you see, the more dots you see here, the more sequences we have from recent times, and you can see this increase. We're seeing more and more reporting of variants of concern, and we're seeing a lot of intermixing between the different states here. So when we think about surveillance of Massachusetts, we have to think about it in the context of the whole region because there is a lot of flow. And we are tracking a number of variants of concern as well. All of these variants that we've heard about from these other places are here already. As of today, there are 359 B117s, 351 of the California identified original variant B1429, and 56 of P1. I particularly want to just take a moment to talk about P1. You may remember it because it was detected first in Manaus, Brazil, in the biggest city in the Amazon region of Brazil. And that region was devastated early on in the outbreak, where it almost was believed to have reached herd immunity. So many people were infected, and then it went away for some time, but then came back in late 2020. At the same time, this P1 mutation had gone from essentially near zero to almost fixation, and is believed to be the kind of origin of this reemerging here. We are now seeing it in Massachusetts. We just the state of Massachusetts just announced a number of cases coming in, 44 and 52 cases of P1 as of today, and 44 in one single transmission cluster. And so the state is tracking this closely. We at MassCPR are working with them closely. This is the kind of case investigations, and it's one of these things where cases emerge, you have to look everywhere, but we want to focus on those places where these new, more concerning variants emerge. We really want to double our efforts there. And we also want to look for new emergent variants, looking for, up here in these charts is looking at increase in likely transmission, or the increase in prevalence of variants. Over, the further to the top and the right, the more likely to be kind of increasing. Some of the things we know about are there, but there are other ones as well. And so we have to track these new variants. And the last thing I'll just say is that, it's really important to be able to see this information in real time. We've been partnering with Fathom Information Design, developing a dashboard for the first time to be able to analyze all of the genome simultaneously and be able to explore this data. So this is all 400,000 genomes in GISAID that are available, and you can explore and look at all of the different mutations, the B117, see where it is, look per country, per variant. And we have a number of different ways of exploring this data, but it's trying to bring tools to the community so that the experts can look at the data the way they need to and find which variants they're interested in, and to explore it in real time and look at new mutations that are popping up in the background. So hope to collectively work on this and sort of, and the last point is, we have to stay vigilant. CDC says this is a real threat to vaccine progress. This is the important thing that we all need to collectively work on. Thank you.
Thank you all. Those were 5 fantastic talks. We're going to move into a question and answer session now. We have about 15 minutes left. We will get through as many questions as possible. So we'll take questions from the Q&A. Feel free to add them, and we will just get through as many as we can. So I'll start and pose a question first to Andrea. This question is, people cannot choose their vaccine. Moderna has announced that a third vaccine dose is being developed against B1351. Do you think that this boost can be used independently of what vaccines people were previously vaccinated?
Yeah, so that's a good question. So the clinical studies, of course, are done with use on people that received the 1273 previously. I think that the vaccine, the new vaccine that's been developed will work well also on people that have received other vaccines or people that were previously exposed to the virus. However, this has to be shown clinically, and I think that this is something that we will need to do in collaboration with other companies and NIH.
Thank you, Andrea. So I've got a question for Katy. So Katy, in the J&J vaccine trial, protection peaked at 71 days, but the data for protection is reported at day 28. The FDA showed that there's 95% protection at day 56, which kind of jives with what we're seeing reported for Pfizer and Moderna that are reporting efficacy at 5 to 6 weeks. So can you comment on whether you think the timing could change the way we interpret vaccine efficacy in the J&J trial? And if I can add one other question there is, do you think that it's something about the length of spike expression that's different across the platforms that could essentially shift, when we're reading out efficacy and why 1 and 2 shots might be different?
So I think it's a really good point about the efficacy going up over time. So if you can see, there's some good figures out there where you can see that drifting up over time, that number. But I think the question highlights a bigger issue, which is that it's really hard to actually compare numbers from one trial to another because there's a lot of differences. So that's one of the big ones, like at what point do you actually measure the efficacy? Day 28 was picked. You had to pick a number at the start of these studies. Maybe a different number would have been better, but that's not the only difference. There's the region, the demographics, and then the force of infection. So how much virus is circulating at the time of the study. So my gut is telling me that the numbers are probably closer to each other in reality, and that it may be more of a numbers difference than something about the platforms being different. As far as I know, I haven't seen any data that compares the persistence of expression between the different vaccine platforms. So it would be totally speculation as to whether that accounts for the difference.
Okay, the next question, it's sort of a two-part question. I'm combining 2 questions to try to get as many as possible, both for Nir. So can MS1 numbers provide predictive value for clinical course? Are they similar to regulatory T cells? And also, can the severity of COVID disease correlate with the intensity of cytokine storm and inversely with MS1 numbers?
Yeah, so thanks for the questions. First of all, in terms of the cytokine storm, yes, it is very correlated with the severity. IL-6 and IL-8 in particular are the ones we find most associated, as well as some chemokines. But the MS1 cells are also correlated with disease severity because they're part of the negative feedback pathway. And so I didn't show it today, but IL-6 is actually the cytokine that induces their formation in the bone marrow. So yes, they're all correlated, both the positive and negative effects are correlated with the severity, and there is some predictive value as well, but they get stronger over time. And they are much more suppressive than regulatory T cells because they suppress innate and adaptive immunity, but there's certainly some similarities, but the mechanisms are not well known at this point.
Thanks, Nir. So I've got a 2-parter for Mandy as well. So Mandy, for the animal models, 2 interesting questions. The first is whether or not animal models may help us understand some of the complications we're seeing from vaccine deployment globally. We're learning a lot about clotting problems with some of the platforms. And so one question is whether that is potentially a platform to understand. And the second is in the context of the non-human primates or the hamsters, is there any evidence that long COVID occurs in those animal models following challenge? And is that another opportunity to begin to understand the pathological events that can help inform how we move out of this pandemic after COVID?
Okay, so for part 1, I do think that, so first of all, the non-human primate would probably be the best place to try to understand some of these adverse events for vaccination. In terms of clotting disorders, maybe, we see early clotting and hyperthrombotic state in the non-human primate model, but we haven't seen any evidence of any kind of massive thrombosis that would lead to the adverse effects that have been reported in people. And I think those are so rare in numbers that it would be almost impossible to model that in an animal study. And this is actually going back to some of our Zika work. I mean, it's very hard to detect low rate events in animal models because you just cannot possibly employ enough monkeys to cover that. However, for the long COVID, there have been some proposals that some of the animal models, including the hamsters, could be a model of long COVID. I haven't seen any convincing data yet on that, but I do think that that as people start to do longer term studies, most of the studies have been very short, up to 14 days maximum in most cases. I do think that we might be able to use the hamster model to look at some long-term effects once people start doing those on a regular basis.
Okay, and multi-part question for Pardis. So first question is, are the tools that you show to look at the variance distribution, are those publicly available, or where is this information available to the general public? Pardis, you're still
Yeah. I actually just answered those 3 questions that are about that I was just typing away, but yeah. So the dashboard that we just showed, we're just finalizing it for release, and it'll be available in basically 2 to 3 or 4 weeks. We're just it's ready, but we want to make sure it's robust before it gets released. But the group that's developing it are fathom.info, and they're long-term collaborators of ours. In the short term, if in general, actually, if you're interested in the variants, there's a website called covariants.org that is a great place to get information about what's circulating right now.
And then the other part to the question, this is more the scientific question, can you speak to the potential of ring vaccination regarding the detection of the P1 variant in Massachusetts? Do you think this is the way to go?
I mean, I think that in general, and thanks, Bruce, for that question. I was just about to answer that one and type that answer. But essentially, I think that there are many reasons why it's useful to know information about variants of concern. But one is that public health, as we know, is understaffed. We don't have enough resources to do everything. And so when you do identify a variant of concern and you see it show up somewhere, and particularly if it's circumscribed, it's a good time to say, let's take our resources and focus there. And that's what the Department of Health is doing. They're really circling around this community where where this major cluster was, and trying to figure out how to support. Ring vaccination in general, when you see something like that, is the right thing to do. I think if I got it, I heard Andrea talk about the studies they'd done with B117 and others. I hadn't heard about P1. So obviously ring vaccination is good if the vaccines work for that particular viral lineage. My guess is that at the very least you should start, and hopefully it is robust and effective to that P1 lineage.
Okay, thank you. So in the last 5 minutes, we'd like to just pose a general question and ask each of the speakers for no more than a 1-minute response. And then apologies for the questions we weren't able to address. Please do take a look in the Q&A and type answers if you can. So the final question for all the speakers is, we've come a long way. In fact, historically, there's never been such progress on vaccines and pathogenesis as what we've seen over the last 12 to 14 months. So what's next? What's the most important questions, and how will those be answered in vaccines and pathogenesis? So Andrea.
Well, I think from my perspective, the most important is to be prepared for future pandemics, and therefore do work ahead on virus and bacteria that can cause a pandemic in the future, and do preclinical studies to be ready for the next wave, because it's going to happen at some point.
Katy.
I think what we really learned from this pandemic has more to do with equity and social and economic differences between populations than anything to do with science. So I think what hopefully in the future we can build for is to improve our public health infrastructure and to make sure that we get vaccines out to everyone around the world who needs them, because in the end, that's going to be more important than any discovery that any of us can do.
Here.
Yeah, in terms of the pathogenesis, I mean, we realize with COVID that it just is so different than the previous viruses that we have to be ready with approaches to study, to create model systems, and to analyze these data sets quickly and work on them to find mechanism. It's been very, very challenging to do that, given how different the symptoms and effects are of this disease. So one of the hopes I have is that we'll find some unifying principles as a result of this intense study in the last year of COVID. That's going to basically give us something we can go after that's more general for severe infections.
Andy.
Thanks. I personally think that we really need to focus on therapeutic interventions now. We have vaccination, but we need to be prepared for when vaccines don't work great. And then, of course, we need to have the second generation vaccines ready to go. I do think this is going to be like the flu one day, where every year we're going to have to get a new COVID vaccine. But I do think we need to have things ready for when people aren't vaccinated and get sick.
Pardis.
Yeah, I'll kind of speak to a little bit of what Andrea said and what Katy said, right? That this is true, that this is not going to be our last pandemic, and we have to be prepared. And we're not done yet, and we may not be done for a while if this virus continues to mutate. And ultimately, I think it is really important that we understand that we are all connected, and if we leave behind any one country, the virus can mutate and change there and come back at us. So it is around, this is a collective effort, and no place in which it is important to take care of each other more than anything.
And I'm going to throw it back at Dan. So I'm going to ask Dan, what is the most important question on your mind today as we hear all about these wonderful scientific advances?
Well, I think that what we've heard today is really just astonishing progress. Progress at a light speed pace that we've never seen before, not even close, in terms of developing multiple vaccines in just a year. And the concomitant advances in immunology, virology, and pathogenesis that goes along with that. But we can't spend any time resting on our laurels, because there's still the majority of people in our country that are not yet vaccinated, and the vast majority of people in the world not yet vaccinated, and the emergence of viral variants that potentially can threaten vaccine efficacy. So I think that, in my view, the most important thing is to accelerate the vaccine campaigns, both in the United States and throughout the world. As we focus on a lot of social justice and equity in the United States, which Katy brought up, which I think is absolutely critically important, we also have to realize that this is a global problem, a global epidemic. I think Pardis brought that concept up. And if we have any region of the world that remains unvaccinated, then that threatens the stability of moving towards resolution of the pandemic everywhere, because there could be new variants that emerge in any region of the world that potentially could be even more problematic than the ones we have today. So we have to realize that this is a global pandemic and requires global solutions.
Thanks, Dan. So with the last minute, I just want to thank all the speakers. I also want to thank MassCPR for their convening activities. I think the one thing that is clear is collaboration has helped to move this entire scientific agenda forward. And so we hope that as we exit this pandemic, we continue to work together as a community. And thanks to the stewardship of the MassCPR. And thank you to the audience for the questions, and have a nice afternoon.
Thank you all.